Hello everyone, welcome back to my channel, Life Sciences by Tumi Silani. This will be a continuation on the 7-part series on the DNA Code of Life. Remember to like, comment, subscribe, and share the link to this channel. My name is Tumi Silani. I am a teacher at Milner High School here in the Northwest Province. Like I said, this is a continuation of the lesson series on the DNA Code of Life. This will be lesson 6 on protein synthesis and amino acid coding. This is a 2-part lesson. The first one will be on protein synthesis, and the second one will be on on amino acid coding. So let us go into the breakdown of the lesson series just to give you a reminder as to what lessons we have done and what lessons we are going to do. On the previous lessons, we have already dealt with Lesson 1, revision of the structure of the cell. Lesson 2, DNA structure. Lesson 3, we dealt with DNA replication. Lesson 4, dealt with the history of the discovery of DNA. The previous lesson, which was lesson 5, we dealt with the RNA structure. And like I said, on this lesson, we will be looking at protein synthesis. It's a 2-part lesson. The first part, we will be looking at the process of protein synthesis, which consists of transcription, movement of mRNA, as well as translation. On the second part of the lesson, we will be looking at amino acid coding. On a separate video, we will be looking at the last lesson, which is lesson 7, which is on DNA profiling. It is a very interesting lesson. We will be looking at the definition and uses of DNA profiling, factors that affect the reliability of DNA profiling. We will be doing some interpretation of DNA profiles, where we'll be determining paternity, as well as looking at forensic evidence in determining a possible criminal in a court case. Moving directly into the lesson, part 1 of the lesson will be protein synthesis. We are now looking at the introduction of protein synthesis. Okay, now, what is protein synthesis for 2 marks? Protein synthesis is a process whereby the cell makes proteins, okay? The word synthesis simply means to make or to manufacture, all right? And in this case, what are we making? We are making proteins, all right? Now, when we look at the definition of protein synthesis, it is the process whereby the cell makes proteins. As simple and sweet as that. Okay. Here is just a note explaining what proteins are and what they do. Proteins are important compounds in the bodies of organisms. Some proteins act as enzymes to catalyze chemical reactions in the body. Remember, you dealt with catalysts in grade 10, while others are structural components of other compounds and structures in the body. Some proteins, like melanin and hemoglobin, are pigments that serve a specific function in the body, while others may act as hormones to regulate and facilitate certain processes. Okay, this was just a note as to what proteins are and what are some of the functions that they may serve. How does protein synthesis occur? All right, now the process of protein synthesis is divided into 3 parts. Transcription, movement of mRNA, as well as translation. Okay. Protein synthesis mainly takes place in 3 successive processes. You must be able to describe and show a detailed understanding of the sequence of events taking place in each sub-process, as well as identify structures involved therein. Okay, now with each process, you are expected to study it theoretically, meaning you need to memorize the steps, and then you must be able to visually study it as well, look into the different structures of the cell where each process takes place, and you must be able to identify and explain the occurrences that happen in each step. Okay, now, before we move on to the process of protein synthesis, there are a few things that I'd like for us to look at. Firstly, a polypeptide chain. When we talk about a polypeptide chain, we are talking about a chain of amino acids. Amino acids are monomers. Here's that word again, monomer. Remember, we said the word monomer means building block. Okay, it is a single unit, it is a single building block that builds a large molecule in the body. Now, proteins are made up of chains of amino acids, which are called polypeptide chains. Okay, now, like I said, the word monomer means a building block. So for us to build a protein, we need to stack amino acids next to one another to build a polypeptide chain, and eventually the cell will process this polypeptide chain into a particular protein. The amino acids are joined together by what we call peptide bonds in a specific sequence to form a polypeptide chain. Please, guys, a peptide bond, polypeptide chain. The word poly means many and peptide means amino acid. Okay, so a polypeptide chain is a chain of many amino acids. Poly, many, peptide, amino acids. Okay, now these amino acids are joined together in a specific sequence. I will explain what do we mean by this when we check the structure of a polypeptide chain. And in between each amino acid, we have what we call peptide bonds. Do not say polypeptide bond. There is nothing such as polypeptide bond. It is a peptide bond. And do not confuse the peptide bond with the weak hydrogen bond. Remember, the weak hydrogen bond is a bond that join two strands of DNA together. So, weak hydrogen bonds are found between the two strands of DNA. However, peptide bonds are found between amino acids in a polypeptide chain. Okay. Now, let's look at the structure of a polypeptide chain. Remember, we said that before a protein becomes a protein, it is first a polypeptide chain. Some people refer to a polypeptide chain synonymously as a protein. Okay, that's fine. So a polypeptide chain is a preliminary stage before a protein becomes a protein. So a polypeptide chain will have an amino acid, then a peptide bond that joins another amino acid, then another peptide bond that joins another amino acid, then another peptide bond that joins another amino acid, another peptide bond that joins another amino acid. So these are the names of the amino acids in this particular sequence. We have meth, ala, tript, lyse, and gly. You don't need to know the names of the amino acids. There are over 30 different types of amino acids, guys. You don't need to know the names of the amino acids in grade 12. You will always be given the names of the amino acids. Most times, you will be given a table that has the amino acids. So don't bother yourself memorizing all the names of the amino acids. It is not necessary in grade 12. Okay, now let us go back. We said that a polypeptide chain is a chain of many amino acids. Remember, poly means many. Many what? Peptides. And peptides are amino acids. Now, these amino acids are joined together by peptide bonds. Okay, guys, so peptide bonds are bonds that are found between the amino acids. They join the amino acids together in a specific sequence, okay, to form a polypeptide chain. And this polypeptide chain will be processed by the cell to form what we call a protein. Now, for us to make a specific protein, the amino acids must follow a specific sequence. So, for example, the amino acid the protein that we are making in this instance must have the recipe of amino acids in a specific sequence, meaning the first amino acid must be meth or methionine, the second one must be ala or alanine, the third one must be trypt or tryptophan, the fourth one must be lyse or lysine, and then the fifth one must be gly or glycine. Okay, guys, this is the specific sequence that must be followed in order for a specific protein to be made. If one amino acid is replaced by another amino acid, a different amino acid, the sequence will change. And if the sequence of amino acids change, it is more likely that a different protein will be made by the cell, or that sequence may not even code for a specific protein. All right. Polypeptide chains will be processed by the cell and eventually become a protein. Therefore, a protein will first become a polypeptide chain and later become the required protein. Okay. To emphasize this point, let us look at the next slide, which has a question and answer activity nyana for us to be able to understand this concept. All right, now here's a question. What if a wrong amino acid is joined together? What happens if a wrong amino acid is joined to the polypeptide chain? What happens if a wrong amino acid is joined to the polypeptide chain? Let us check. Here we have the original polypeptide chain having the correct sequence of amino acids. Let us just number them. 1 must be meth, 2 must be ala, 3 must be trypt, 4 must be lyse, and the 5th amino acid must be gly. Okay, guys? This is the specific sequence of amino acids in our polypeptide chain that will give us the required protein, the protein that the cell wants to make. However, the question is asking, what happens if a wrong amino acid is joined to the polypeptide chain? Let's look at this mutated polypeptide chain. Now, in this polypeptide chain, the first amino acid is meth, the second amino acid is ala, that is correct. Third one is trypt, that is also fine. But on the fourth amino acid, let's check. Arg replaces lyse, okay? And on the 5th amino acid, the 5th amino acid is actually correct as well, which is gly. Okay, now we have a mutation that occurred here. We have an error that occurred here. Lyse is now replaced with arg. This is a wrong amino acid It was supposed to be lice on the fourth one. However, now that is replaced with arg. Okay, as you can see, if a wrong amino acid is included in the polypeptide chain, the sequence of amino acids is going to change, and if the sequence of amino acid changes, then a wrong protein or a different protein will be made. Okay, guys? Now, the sequence of amino acids in our polypeptide chain to make the required protein needed to be meth, ala, tript, lyse, and gly. Now, if lyse is replaced by arg, there is going to be a different sequence of amino acids. So instead of meth, ala, tript, lyse, and gly, now we will have meth, ala, tript, arg, and gly. Okay, guys? Now, what is going to be the effect on the protein that was meant to be made by the cell? The sequence of amino acids will change, as we can see there, and as we have explained. Then a wrong protein will be made, or sometimes no protein will be made. Okay, so this is the original polypeptide chain, which contains the required sequence of the required sequence of amino acids to form the required protein. However, if a wrong amino acid is included, then the sequence of amino acids will change, then a wrong protein or a different protein will be made. Sometimes the sequence is so muddled up that no protein will be made at all. All right. This may result in a gene mutation. We will talk about gene mutations when we deal with genetics in the second term. Okay, with that background information, we are ready to deal with protein synthesis. Protein synthesis is divided into three parts: transcription, movement of mRNA, and translation. Remember, it is your job to know the things or the occurrences that happen during transcription, during movement of mRNA, and during translation. We are going to learn about this both visually and theoretically. So it is important for you to memorize the steps of transcription, mRNA, and translation, and understand those steps in terms of looking at different visuals and being able to explain those visuals. Let's get right into it. Right, protein synthesis. The first part of protein synthesis is called transcription. What is transcription? Transcription is the process where mRNA is made, 1 mark, from a DNA template, 1 mark. Remember what we said a template is. We said a template is a foundation where you are going to stack things on top to make something else. Just like a slice of bread is a foundation where we stack different ingredients like cheese, margarine, whatever, in order to make a sandwich. Okay, so during transcription, mRNA, messenger RNA is made, okay, but that is made from a DNA template. We will see how that happens. Second question, what is the site or the location of transcription? And transcription simply occurs inside the nucleus. All right. Right, now let's look at the steps of transcription. We are now describing the process of transcription. For you to understand transcription, it is important for you to have memorized the steps of DNA replication. Transcription and DNA replication are similar. They are not the same because remember, in DNA replication, we are making an identical copy of DNA. However, here we are making mRNA from a DNA template. The steps are similar, but there are key distinguishing elements that differentiates DNA replication as well as transcription. Okay. So we will be using this visual to explain what transcription occurs. As you can see over there, we have DNA as a double helix, and at that part DNA is separated, and then that strand of DNA there, as you can see, acts as a template to make this guy over there, which is mRNA. Remember what we said transcription is? We said transcription is the making of mRNA from a DNA template. So as you can see over there, there is a DNA strand that is acting as a template to form that strand over there, which is mRNA. Okay, now let us look at the steps of transcription. Step number 1, the DNA double helix unwinds. Remember, DNA is a double helix, meaning it has two strands, strand 1 and strand 2, and both those strands are twisted. It is impossible for DNA to make mRNA if it is still a double helix. So the first step is that DNA double helix will unwind. It will become straight. Okay. Step number 2, the weak hydrogen bonds break. Remember, the two strands of DNA are joined together by weak hydrogen bonds. All right? Now, after the DNA double helix unwinding, the weak hydrogen bonds that are joining the two strands of DNA break. After the weak hydrogen bonds break, then two DNA strands separate. As you can see, after the weak hydrogen bonds have been broken, that strand separate and that strand also separates. So they separate. Or you can say that they unzip. You only need to say one. You can say two DNA strands separate or two DNA strands unzip. You don't need to say both. Then only one strand acts as a template. Okay, now check this visual there. Which strand, the left strand or the right strand, is making mRNA? It is actually that strand there, the right strand. So, the DNA double helix unwinds and the weak hydrogen bonds will break. The two strands will separate and only one strand acts as a template. Remember when we were undergoing the process of DNA replication, we said both strands act as a template. However, in transcription, only one strand acts as a template. So in this case, it is the right strand that is acting as a template. Just to remind you, a template is a strand on which we are going to build something. We are going to stack things on top to build something. In this case, it is a template to build an mRNA strand or an mRNA molecule. Step number 4, free-floating RNA nucleotides from the nucleoplasm attach to the template in a complementary way. In a complementary way meaning adenine will bond with uracil and cytosine will bond with guanine. Remember, complementary base pairing in RNA, A bonds with U, not T, guys, not thymine. Remember, RNA contains uracil. It does not contain RNA. So, if we have a strand, for example, if we have a strand that acts as a template, the DNA strand, if it has T, A, G, and A, G, G. Let's say, for example, this is our template strand. This is our template strand. And it is a DNA strand. Why? Because it contains thymine. All right? Now, free-floating RNA nucleotides are going to come and attach themselves to the complementary strand. The nucleotides will contain complementary bases to the DNA strand. Okay? So in RNA, T on the DNA template will attract a RNA nucleotide containing nitrogenous base A. Okay? And as you can see over there, we have A on the DNA template. It will attract a nucleotide containing a nitrogenous base U. And then G on the RNA strand, it will be C. Okay? And then A on the RNA strand will be U. And then G on the RNA strand will be C. And then G on the RNA strand will also be C. Meaning, this is our DNA strand. How can we confirm that it is a DNA strand? Because it contains T. It contains thymine. So this is our DNA template. From this DNA template, we are making an mRNA molecule. Okay? This is an mRNA molecule. Why? Because it contains the nitrogenous base U. So there we have uracil, and there is another uracil there. Okay? So from a DNA template, from a DNA template, we are making an mRNA strand or an mRNA molecule. So how do we do this? After the DNA double helix unwinding, the weak hydrogen bonds breaking, two DNA strands separating, and then only one strand acting as a template, free-floating RNA nucleotides. Please do say RNA nucleotides from the nucleoplasm attached to the template in a complementary way. So all these free-floating RNA nucleotides will come and attach themselves and attach themselves and attach themselves to the template in a complementary way, like I've explained there. Then the last step, one mRNA molecule is formed carrying the DNA code for making a protein. Remember, guys, let me just erase there. Remember, DNA is a let me just make a circle there. DNA is a nucleic acid that is found inside the nucleus. DNA is found inside the nucleus. Nucleus. Excuse my handwriting. I am trying to write using a mouse. I'm not using a pen. Okay, so this is a nucleus. This is the nucleus. DNA is too large to leave the nucleus. DNA is too large to leave the nucleus. Now, what DNA does is that DNA contains a code in terms of which protein needs to be made. So the instructor of which protein needs to be made is DNA. DNA instructs the cell as to which protein needs to be made. And the instruction is carried in the sequence of nitrogenous bases on the DNA. But the proteins are made outside the nucleus in the cytoplasm at the ribosomes. So outside the nucleus, we are going to have ribosomes. But because DNA is too large to move out of the nucleus, it requires mRNA. The word mRNA stands for messenger RNA. So what messenger RNA does is that it takes the DNA code for which protein needs to be made and transfers this DNA code, takes it to the ribosomes where the proteins are going to be made. Now, mRNA is called messenger RNA. Why? Because it contains the DNA code to tell the cell which proteins is to be made there at the ribosomes outside of the nucleus. So the instruction comes from DNA and it is written down, or it is transcribed into the messenger RNA, and then the messenger RNA takes this transcribed code in terms of the sequence of nitrogenous bases. It takes it to the ribosomes, and the protein, the required protein, will be made at the ribosomes. Hence, step number 5 is one mRNA molecule is formed carrying the DNA code for making of a specific protein. Okay, on the next slide, we will be looking at a visual explaining this. On this slide, we have a visual explaining what we are talking about. So there is the cell. Inside of the cell, we are now inside the nucleus. Okay, now there is our ribosome, and like we said in the past, when we uncoil not a ribosome, actually a chromosome. When we uncoil a chromosome, we are going to find DNA. Okay, so remember, we are explaining the process of transcription. So I will be playing the video and pausing, and it will explain the different steps of transcription. Remember, guys, it is your job to check and memorize each and every single step, sentence by sentence, word by word of transcription, so that you will be able to affiliate those steps to the visuals that you will be given in a given question paper. Okay, there we have our DNA. And then we said that step number 1, what happens? The DNA double helix unwinds and the weak hydrogen bonds are going to break. Now, this does not spontaneously happen. All the steps of transcription are controlled by enzymes. As you can see over there, that enzyme is called RNA polymerase. You don't need to know the names of the enzymes that control the different steps of transcription, and otherwise, just know that each step, everything that happens to the DNA and RNA and the nucleotides are controlled by enzymes. They do not spontaneously or magically happen. Okay, so DNA double helix unwinds and weak hydrogen bonds break. Let's check. Let's check over there. The part that is transcribing, do you see there? The DNA is still a double helix, but then the part that is transcribing will unwind. So that part becomes straight. Okay, guys? Now, DNA double helix unwinds, and then we said that the weak hydrogen bonds are going to do what? The weak hydrogen bonds are going to break. So on that part over there, as you can see, the weak hydrogen bonds are breaking. And then when the weak hydrogen bonds break, what is going to happen to the two strands? The two strands of DNA are going to separate. So there they are separated. Okay, now we said, though we have two strands of DNA that are separated, only one strand is going to form a template. Okay, in this case, the strand that is forming a template is the bottom strand. Step number 3, we said that free-floating nucleotides. There we have a nucleotide, there we have a nucleotide, there we have a nucleotide, there we have a nucleotide. Remember, this is occurring inside the nucleus of a cell. So we said free-floating RNA nucleotides will attach themselves to the template in a complementary way. And this is exactly what is happening. I'm going to pause again, and you must check what is happening. There we go. The free-floating RNA nucleotides are attaching themselves, as you can see, to the templates in a complementary way. By complementary way, what do we mean? We mean that if on the DNA template we have thymine, a RNA nucleotide containing nitrogenous base A will bond there. If we have C on the DNA template, a free-floating RNA nucleotide containing nitrogenous base G will bond there. So if you can check, all the free-floating nucleotides, RNA nucleotides, are attaching themselves in a complementary way. Okay, and this is a DNA template. Remember, this is the coding strand. We call it a coding strand because it is the one that is acting as a template to build the mRNA. That one is just chilling. The other one is not chilling. It is a non-coding strand. It is not coding. Nothing is happening to it. So only the template will be coding for the mRNA. Okay, guys? Let's check. So the free-floating mRNA nucleotides are attaching themselves to the template in a complementary way. Excuse me. Let me just go back. As you can see over there. Okay. So as they are attaching themselves to the template, to the DNA template in a complementary way, they are building an mRNA strand. There they are, attaching and attaching and attaching and attaching and attaching. And step number 5, we said that one mRNA strand will be formed, and this mRNA strand will contain the DNA code will contain the DNA code for which protein is going to be made. Okay, guys? Now, that is the first part of protein synthesis, which is known as transcription. Which brings us to an activity. I am pausing the lesson and we are going to an activity. So, on this activity, you are supposed to tabulate 3 differences between DNA replication and transcription. Remember, I said to you, DNA replication is quite similar to transcription. However, there are key elements that are different between DNA replication and transcription. You need to memorize the steps, guys, line by line, sentence by sentence, word by word. Okay, now, remember the term tabulate. The term tabulate means put in a table. So the first thing that you must do is that you must draw a table. And remember, it says tabulate 3 differences between DNA replication and transcription. Meaning, on the headings of your different columns, you must include these precise words: DNA replication and transcription. There we go. Okay, now let us tabulate. Now I'm going to be comparing step by step, and I am expecting you step by step to compare DNA replication to transcription. Step number 1, during DNA replication. Step number 1, DNA double helix unwinds. Step number 1 in transcription, DNA double helix unwinds. So there is no difference in step number 1. Step number 2 of DNA replication, weak hydrogen bonds break. Step number 2 of transcription, weak hydrogen bonds break. So there's no difference there. After the weak hydrogen bonds break, what's going to happen? The 2 DNA strands will separate during transcription, and during DNA replication, the 2 DNA strands separate. So there's no difference there. The difference comes in here. During DNA replication, only one, okay, excuse me, let us skip that. Both DNA strands acts as a template. Let's skip the first one. Let's skip that one. I'll come back to that one. Both DNA strands acts as a template. However, in transcription, only one strand acts as a template. Okay. And then the following one. DNA nucleotides attach themselves to the template in a complementary way. However, in transcription, RNA nucleotides attach themselves to the template in a complementary way. Okay, and essentially during DNA, at the end of DNA replication, DNA makes an identical copy of itself. During transcription, the cell makes mRNA. Okay, or you can say during DNA replication, DNA makes two DNA molecules, but during transcription we are making what? We are making an mRNA strand or one mRNA molecule. As for the marking guideline, let me just erase, please. There we go. How do we mark? Remember, like I said in the previous lesson, the way that we mark this, you will see your marker writing the letter T, like that. T for the table. But remember, I advised you, this T is not for just drawing the table. This T is for including the relevant headings of the columns of the table. On one side, we have DNA replication. On the other side, you have transcription. If you have that, then you will see your marker will put a tick. If you have wrong variables as your headings, your marker will just make a cross. Okay, so it is 1 mark for the table, and because the question said tabulate 3 differences between DNA replication and transcription, you are going to receive a mark for any of the first 3 corresponding differences. Okay, 1 mark, 1 mark, 1 mark on the DNA side, and 1 mark, 1 mark, 1 mark on the RNA side. The questions for tabulation is always an odd number. It can be 3, it can be 5, it can be 7, it can be 9. It is never an even number. It is never an even number. Why? Because there's always that 1 mark for the correct table. Okay, guys? Okay, just to caution you again, guys, mind the following questions. First question, tabulate the differences between the structure of DNA and the structure of RNA. This is not the same question as tabulate the differences between the structure of a DNA nucleotide and an RNA nucleotide. This question, you are only going to give the differences in the nucleotides of DNA and in the nucleotides of RNA. It is not an overall structural difference. So here you may say that DNA is long and RNA is short. DNA has 2 strands and RNA has 1 strand. Okay, and you may include the differences in the nucleotides of DNA and RNA. So this question is a bit more general. However, this question over here, you are not going to include the length of DNA and the number of strands, because it is requiring you to only give the differences in the nucleotides. So the differences in the nucleotides is that DNA contains deoxyribose sugar in its sugar portion, and RNA contains ribose sugar in its sugar portion. DNA contains the nitrogenous base thymine, and then RNA nucleotide contains the nitrogenous base uracil. So here it is only the differences in what? It is the differences in the nucleotides. Then here is a bit more open. You can give any structural differences between DNA and RNA. Do not confuse these two questions again with the following question. You might be given a question that says, tabulate the differences between transcription and DNA replication. Okay, so this is the differences in the processes, transcription and the process replication. So this question is requiring you to give the differences in the processes, not in the structures. Not in the structures, not in the nucleotides. So be very careful about these three questions, because it is very easy to make mistakes and lose marks. Okay, guys, I'm moving on. We are now continuing with protein synthesis, and we will be looking at movement of mRNA. We will be looking at movement of mRNA, which is the second part of protein synthesis. Now, after mRNA is made inside the nucleus through the process of transcription, what is going to happen to the messenger RNA? mRNA moves from the nucleus to the cytoplasm through the nucleopores. Remember, the nucleopores are pores or holes that are on the nuclear membrane. And then it is through the nucleopores that mRNA is going to move from the nucleus and is going to move to the cytoplasm. Just a note, like I said in the previous slide, DNA is too large to fit through the nucleopores. It is trapped inside the nucleus permanently. Hence, it requires a messenger to pass the instruction to the ribosomes. The ribosomes are where? The ribosomes are in the nucleus, for which protein or proteins need to be made. During transcription, the DNA code in the form of the sequence of nitrogenous bases is transcribed. The word transcribe means written down onto the mRNA strand. The mRNA then moves out of the nucleus and passes this code to the ribosomes, where the required protein will be made. Therefore, DNA provides the code for which protein is to be made. mRNA then takes this message to the ribosome, where the protein is to be made. Okay, so the step is movement of mRNA, and then you will only write mRNA moves to the nucleus. Moves from the nucleus to the cytoplasm through the nucleopores. That's the answer. This part over here, this note over here, is explaining the role that DNA takes in protein synthesis and the role of messenger RNA in protein synthesis, which might be a question. You might be asked, provide the role that is played by DNA or provide the role that is played by mRNA in the overall process of protein synthesis. The third part of protein synthesis is known as translation. Just like the first part, transcription, and the second part, movement of mRNA, you need to memorize, guys, the steps that takes place here, and you need to learn the process visually. As you will see later when we do activities, you need to associate the different steps to different visuals, different pictures. Okay? Now, translation, the process. Okay, firstly, the location. Where does translation take place? Translation occurs at the in the cytoplasm at the ribosomes. In the cytoplasm at the ribosomes. Please write it down in full. In the cytoplasm at the ribosomes of the cell. And this is outside the nucleus. Guys, Remember, the different parts of the cell we learned about in lesson 1 on this video series. If you do not understand the different parts of the cell, if you don't understand where the cytoplasm is, what are the different parts of the nucleus, where are the ribosomes, I suggest that after this lesson, you go back to lesson 1, the video on lesson 1 on this playlist, and then you look at the revision of the structure of the cell, because it will be very problematic for you to associate all of these steps of protein synthesis if you are not well versed in the different parts of the cell. Okay, so the location where translation takes place is the cytoplasm at the ribosomes, and this is outside of the nucleus. Right. Description of the process. The first thing that mRNA does when it reaches the cytoplasm is that it attaches to a ribosome, 1 mark. The second type of RNA that we're dealing with, tRNA. tRNA molecules bring specific amino acids. Specific amino acids, not a random amino acid, but specific amino acids to the ribosome, another mark. According to the sequence of codons of the mRNA, which are complementary to the anticodons of the tRNA. Peptide bonds join the sequence of amino acids to form a polypeptide chain. The polypeptide chain will be processed by the cell to form the required protein. This point does not carry a mark. Now, the first thing that mRNA does is that it attaches to a ribosome, and then tRNA molecules bring specific amino acids to the ribosomes according to the sequence of codons of the mRNA, which are complementary to the anticodons of the tRNA. Then the peptide bonds join the sequence of amino acids to form a polypeptide chain, and this polypeptide chain will be processed by the cell to form the required protein. Now, I have introduced you to the following things in this description: a tRNA molecule, codons on the mRNA, as well as anticodons on the tRNA. We will look at that in the next couple of slides. For now, you need to pause and memorize these steps. You can write them down on a piece of paper. You need to memorize them, bullet by bullet, word by word, sentence by sentence, because you will be required to answer them like this. Okay. On this slide, we are explaining translation using visuals. So the steps that you learned about on the previous slide, now we are going to visually see them here. Remember, we are picking up from transcription, from when we made mRNA. Okay, guys? Now, there is the mRNA. It is leaving the nucleus and it is going to the cytoplasm. It leaves the nucleus via the nucleopores. Okay, that is the second step. The first thing that we said the mRNA does when it reaches the cytoplasm, okay, so this is when translation begins. The mRNA will attach to a ribosome. There we go. So there is our ribosome and there is our mRNA. As you can see over there. Now, the next part says that tRNA molecules bring specific amino acids to the ribosome, okay, according to the codon of the mRNA and the which are complementary to the anticodons of the tRNA. I will explain that part just now. So there we have a tRNA molecule. When you check there is a tRNA molecule. The job of tRNA, the full term is transfer RNA. What is it transferring? One transfer RNA brings one amino acid. It carries one amino acid at the top. So there is the amino acid binding site, and it is carrying an amino acid. So one tRNA carries one amino acid at the top. Okay. At the bottom, you will see that we have three nitrogenous bases. First one, second one, and third one. And the three consecutive nitrogenous bases on mRNA are called an anticodon. The three consecutive nitrogenous bases that are found on tRNA are known as anticodons. So, tRNA brings specific amino acids, okay, according to the codon of the mRNA, which is complementary to the anticodon of the tRNA. Let's check what is happening. So the tRNA is going to the ribosome at the where the mRNA is attached. Let's just wait for it to move. There we go. Let's check what is happening. So each time a tRNA gets to the ribosome, let's check what it's doing. It is bringing an amino acid. Now, let us focus here. I want to explain what an anticodon is versus what a codon is. Now, check over there. Like we said, this is a tRNA. This is a tRNA. At the top, it is bringing one amino acid. At the bottom, it has three nitrogenous bases. These three nitrogenous bases, U, A, C, are called anticodons. And the anticodons must be complementary to what we call the codons of the mRNA. Now, the three nitrogenous bases that we find in tRNA, we said we call that the anticodon. Three nitrogenous bases that we find on mRNA are called the codon. The codon of the mRNA must be complementary to the anticodon of the tRNA, so that the tRNA may come and bond itself to the mRNA. The tRNA that comes to the ribosome bringing an amino acid is not any random tRNA. It must be a tRNA of which its anticodon is complementary to the codon of the mRNA. So, in that way, if the anticodon is complementary to the codon, we know that it is bringing a specific tRNA. The tRNA is bringing a specific amino acid, not just any random amino acid. The amino acid, I'm repeating this again. The amino acid that is brought to the ribosome by the tRNA is a specific amino acid. How do we know which amino acid do we need to bring to the ribosome? It is the amino acid of which its tRNA anticodon is complementary to the codon of the mRNA. What is an anticodon? An anticodon is three nitrogenous bases that we find in the tRNA. What is a codon? A codon is three nitrogenous bases that we find in the mRNA. Okay, guys? So in a way, mRNA will only accept the tRNA of which its anticodon is complementary to its codon. By that, the tRNA will bring a specific amino acid and not a random amino acid. Okay, let us unpause and check. So as the tRNAs, let me just go back. As the tRNAs are bringing specific amino acids to the ribosome, let's check what is happening at the top. Peptide bonds bond these amino acids together to form what we call a polypeptide chain. And this polypeptide chain will be processed by the cell. So it is done, will be processed by the cell to form that. And that we call a protein. That we call a protein. So at first, this protein was a polypeptide chain, a chain of amino acids, and then it will be processed by the cell to make a protein. Okay, guys? So what I suggest you do at this point, I suggest that you go back to the slide where the different steps of translation are laid out. Read, memorize those steps. Come back to the slide, look at and associate each step of the process of translation, and associate each step to the different structures included in this simulation. I can show you the simulation all day. It can run over and over and over and over and over and over and over again. However, if you have not memorized the different steps of translation, if you have not memorized the different steps of movement of mRNA, if you have not memorized the different steps of transcription, even if you see what is happening, you will not be able to understand because you do not know the different steps. So the first thing that you need to do, memorize the steps. After memorizing the steps, associate each step with the simulations that I have provided to you, and then that will make sense. Okay, so in that way, you would have learned the parts of protein synthesis theoretically as well as visually as well. Okay, guys, I am moving on. Okay. On this slide, we are looking at the structure of a tRNA molecule, a transfer RNA molecule. There is our transfer RNA. Now, we are supposed to study what this transfer RNA looks like. At the top part, we have a amino acid, okay? And this is the body of the tRNA. And then at the bottom, we will see 3 nitrogenous bases, and we say that those 3 nitrogenous bases are a anti-codon. So AGA is 3 nitrogenous bases on that tRNA, and we refer to that as a codon. Okay, so there is our tRNA. At the top, it is carrying an amino acid that is called alanine. Remember, you don't need to know the different amino acids by names. You will always be given the amino acids in your question papers. And then at the bottom, that is our anticodon. Remember, the job of tRNA is to transfer amino acids to the ribosomes, specific amino acids to the ribosomes, meaning the anticodon AGA is a codon that specifically, so this codon AGA is a codon, this anticodon AGA, pardon me for that, this anticodon AGA is a specific anticodon that codes for the amino acid alanine. So it codes for alanine and alanine alone. No other amino acid in this instance. So a specific tRNA anticodon codes for a specific amino acid. Now, this transfer RNA will transfer this amino acid to the ribosome. Okay, guys? Remember, on the mRNA, let's say, for example, this is an mRNA strand, you will find a sequence of nitrogenous bases in triplets. 1, 2, 3, 1, 2, 3, and 1, 2, 3, and 1, 2, 3, 1, 2, 3. Okay. Let's say, for example, let's say this is A, C, G, and this is U, A, C, and the likes. Now, what must this codon be? Check this codon. It's complementary to this codon. Must be complementary to this tRNA. So we just need to code. A is complementary to U, G is complementary to C, and A is complementary to U. Okay, so this is the anticodon of the tRNA, and then on the mRNA we find codons. It is 3 consecutive nitrogenous bases on the mRNA molecule. How many codons do we have in this mRNA molecule? We have 1 triplet, 1 codon. We have the second one there. We have the third one there. We have the fourth one there. And then we have the fifth one there. Meaning 1 codon has 3 nitrogenous bases. 1 anticodon has 3 nitrogenous bases. Now, the tRNA that comes to the ribosomes and attaches itself to the mRNA must contain an anticodon that is complementary to the codon of the mRNA. Okay, let us just make another example. Let us take another example. So, here we have the codon on the mRNA that reads ACG. What is the complementary anticodon? A will bond with U, C will bond with G, and G will bond with what? It will bond with C. Now in the blue, we have the complementary anticodon, which is complementary to the codon, the first codon of the mRNA. Let's take another example. The second codon. Let us code for the anticodon of the mRNA, of the tRNA. So U will bond with A, and then this A will bond with U, and then C will bond with G. Do you see? Now, this is the mRNA codon, and this is the complementary tRNA anticodon. Okay, guys? All right. Just some notes over there. Let me just erase to create space. Okay. Each tRNA carries 1 amino acid. So, meaning we have a 1:1 ratio. Okay? 1 tRNA carries 1 amino acid. 3 consecutive nitrogenous bases on a tRNA are called an anticodon. Okay? So the ratio 1 tRNA, 1 amino acid. Okay? 1 codon equals to 3 nitrogenous bases. Each tRNA has one codon. So, one tRNA, one amino acid, one anticodon. Okay? The ratio, therefore, per one tRNA, there is one anticodon. 1 is to 1. And one codon. Okay? Now, on the mRNA, this one anticodon will be complementary to one codon of the mRNA. Per one amino acid, there will be one anticodon. The ratio tRNA to amino acid is 1 is to 1 is to 1. Okay? Let me show you again. Let me draw. Okay. Nope. Let me draw an mRNA strand. 3 nitrogenous bases, 1, 2, 3, and then it will be U, C, U. Let's check. One tRNA carries one amino acid. One tRNA carries one anticodon. Are you with me? So, 1 is to 1, 1 is to 1. Okay? This one tRNA has one anticodon, and one anticodon is complementary to one codon of the mRNA. So we can essentially say one codon of the mRNA codes for one amino acid, because it will draw in one tRNA which carries one amino acid. One codon of the mRNA is complementary to one anticodon of the tRNA. Okay? And one tRNA anticodon is carried by one tRNA, and one tRNA carries one amino acid. The only difference is there. The only difference is there on the ratio of anticodon to nitrogenous bases. That is 1 is to 3. Everything else is 1 is to 1 is to 1. Okay? So if you get a question, for instance, how many codons are required for one to code for one amino acid is 1. How many anticodons are needed for one amino acid is 1. How many tRNAs are needed for one mRNA codon is 1. Everything else is 1. The only difference is that one anticodon is made up of 3 nitrogenous bases. So nitrogenous bases is 1 is to 3. But everything else, the ratio is 1 is to 1. All right. Okay. Now here is an overview of protein synthesis. We said protein synthesis occurs in 3 parts. The first one, transcription, where mRNA is made from a DNA template. The second part, that mRNA that is made during transcription then moves to the cytoplasm through the nucleopores, and that we know as movement of mRNA. And then last but not least, we have translation, where amino acids are joined together at the ribosome to form the required protein. Okay, so this is at the cytoplasm at the ribosomes. this is a brief overview of the three parts of protein synthesis. Okay, with that being said, let us now apply what we have learned into in the following activities. As per usual, this side we will have the questions and this side we will have the answers. Remember, try to work out the answers in a piece of paper before I give you the answer. So, after receiving the question, please pause the video, write down the answer, and then compare that to my answers. If you have a difference in opinion, or if you have something to input, please put that in the comments. I would really appreciate that. The diagram below represents a polypeptide chain. Remember, what is a polypeptide chain? Poly means many, peptide means amino acids. So a polypeptide chain is a chain of many amino acids that will eventually form a protein. Right. So we have how many amino acids in our polypeptide chain? We have the first one, meth, the second one, ala, the third one, tript, the fourth one, lyse, and then the fifth one we have as gly. Okay, remember we are studying the diagram before we even look at the questions. So we have 5 different amino acids in a specific sequence. Okay, they need to be in the specific sequence. Now let's look at question 1. How many tRNA molecules are required to build the polypeptide chain above? How many tRNA molecules are required to build the polypeptide chain above? Remember, we have if you're asked to draw a tRNA, you can just draw any shape. We have a tRNA itself. On top, it is carrying only one amino acid. Okay, guys? It's carrying only one amino acid. And then at the bottom, our tRNA will be carrying an anticodon. And what did we say? An anticodon is 3 nitrogenous bases. So let's say, for example, this is UUA, for example. Okay, so that part, one tRNA, one anticodon, one amino acid. So if we have 5 amino acids, how many tRNAs are required to form that polypeptide chain? 1 tRNA carries 1 amino acid. So the ratio is 1:1. Okay, so we are going to need how many how many tRNAs? We are going to need 1, 2, 3, 4, and 5. Okay, so it says explain. The question says explain. So you need to give details to this. Okay, and when you check the mark allocation, this is a question that carries a lot of marks, 3 marks. Each tRNA carries 1 amino acid, 1 mark. Since there are 5 amino acids above 1 mark, it would take 5 tRNAs to build this polypeptide chain. Okay, second question. Explain how many tRNA anticodons are required to build the polypeptide chain above. tRNA anticodons. Remember, we said that 1 tRNA carries 1 amino acid and 1 tRNA carries 1 anticodon. Okay, so 1 anticodon actually codes for 1 tRNA. Okay, guys, so since we have 5 mRNAs we are going to need 5 tRNAs. This question again says explain. This question again says explain. The mark allocation is actually quite expensive as well. It is 3 marks. So the answer to question 1.2 would be each tRNA carries 1 anticodon, 1 mark, which codes for 1 amino acid, 1 mark. Since there are 5 amino acids above, it would take 5 anticodons to build this polypeptide chain. Okay, let's look at the third question. Explain how many mRNA codons are required to build the polypeptide chain above. How many mRNA codons? All right, now let's check. If this is our mRNA, strand, okay, remember, an anticodon of tRNA is complementary to 1 mRNA codon. So what will be the complementary here? It will be, the complementary codon will be AAU. It will be AAU. These 3 sequences of or consecutive nitrogenous bases on the mRNA are a codon. Now let's check. 1 tRNA contains 1 anticodon. And that 1 anticodon codes for 1 amino acid. That 1 anticodon is complementary to 1 what? Codon of the mRNA. Okay, remember, the mRNA has many nitrogenous bases or many codons. 1, second codon, third codon, then this one is the 4th, and then we have a 5th codon. Now let us check what's happening there. Okay, 1 tRNA codes for 1 amino acid. 1 tRNA has 1 anticodon. If we have 1 anticodon, it is complementary to 1 codon of the mRNA. Okay, guys? Now, how many amino acids do we have? We have 1, 2, 3, 4, and 5. Meaning, how many codons of the mRNA are we going to need? We are also going to need 1, 2, 3, 4, and 5 codons. Okay? Because we are going to need 5 tRNAs that come to the mRNA. So the ratio is 1:1. But we need to explain. And this one is worth 5 marks. The answer, each tRNA carrying 1 anticodon and 1 amino acid. Each anticodon bonds with 1 mRNA codon. Since there are 5 amino acids above, it would take 5 codons to build this polypeptide chain. All right. If you're feeling confused, please go back, rewind, go back, check and study again these numbers, okay? Because these are very common exam type questions. All right, let me just erase so that we can see that question properly. Okay, following question says, calculate how many nitrogenous bases on the mRNA would be required to build the polypeptide chain. Show all workings. Calculate how many nitrogenous bases, not codons, nitrogenous bases on the mRNA would be required to build the polypeptide chain. Show all workings. Okay, guys. Now, one codon contains 1, 2, 3 nitrogenous bases. One codon, let me just write it down. So, one codon codes for, like we've explained in the previous answer, it codes for 1. It codes for 1 amino acid, and 1 codon equals to how many nitrogenous bases? Equals to 3. Equals to 3 codons. 3 nitrogenous bases, sorry. 1 codon equals to 3 nitrogenous bases. I repeat, 1 codon equals to 3 nitrogenous bases. And 1 codon codes for 1 amino acid. Therefore, we have how many nitrogenous bases in 1 codon? We have 3 nitrogenous bases. Okay, guys? We have 3 nitrogenous bases. And how many amino acids do we have in this polypeptide chain? We have 5 nitrogenous, sorry, we have 5 amino acids here. Okay? 1 codon codes for 1 amino acid. 1 codon has 3 nitrogenous bases. So it would be 3 times 5 amino acids, which will give us what? Okay, let us look at the answer to this question. Let me just erase the explanation and let us check. So the answer is 3 nitrogenous bases times 5 amino acids will give us a total of 15. Okay, will give us a total of 15. So there, okay, another mark should be, forgot to add it, another mark should be there because it's 3 marks. So it is 3 nitrogenous bases times 5 amino acids gives us 15. The final answer is 15, and that is going to be our compulsory mark over there. All right. Whew. That's a lot of calculations and understanding. Please, guys, if you feel confused, it's fine. It's totally okay. It's totally okay at this point to feel confused. Just go back, check again what I'm talking about, take notes to digest it, and then look at the video again, look at the explanation again, redo the activity on your own, and then you'll understand it better. Okay, on the following slide, we will be looking at different molecules and their roles in protein synthesis. Remember, we have how many molecules? We have DNA, the first molecule that is involved in protein synthesis during transcription. We have mRNA, we have tRNA, we have a third RNA, which is called ribosomal RNA. We will talk about it just now. And yeah, we have 4 molecules that are involved in protein synthesis. So in the following table, it is a summative table that is going to summarize the roles of each molecule and where do we find it and the likes. Okay, for your convenience, I've put these molecules, their roles, in a table to make it very easy for you to study. So be welcome to take a screenshot or write it down. You will definitely need it when you do your revisions, because you might get a question of what is the role of one or two of the following molecules in your exams. Molecule, site where it is produced, site where it functions, and the role or significance of or function during protein synthesis. The very first one is DNA. Where is DNA found? DNA, remember, is trapped inside the nucleus, so it is found inside the nucleus. Where does it function? In protein synthesis is that okay. For your convenience, I've put these molecules, their roles, in a table to make it very easy for you to study. So be welcome to take a screenshot or write it down. You will definitely need it when you do your revisions, because you might get a question of what is the role of one or two of the following molecules in your exams. Molecule, site where it is produced, site where it functions, and the role or significance of or function during protein synthesis. The very first one is DNA. Where is DNA found? DNA, remember, is trapped inside the nucleus, so it is found inside the nucleus. Where does it function? It functions in the nucleus, okay? And the role that it plays during protein synthesis is that it acts as a template for the formation of mRNA during transcription. That is not the only thing that it does. During transcription, it provides the genetic code for which protein is to be made. All right? The second molecule is messenger RNA, mRNA. Where is it produced? Remember, it is produced in the nucleus during transcription. Okay, remember we said that transcription is a process where mRNA is made from a DNA template. It is made it functions at the ribosomes, which are in the cytoplasm. And then what does mRNA do? It transfers the genetic code from the DNA template to the ribosomes, where the required protein will be made. So it transfers the genetic code from the DNA template to the ribosomes, where the required protein is to be made. So true to its name, messenger RNA, it transfers the message of the genetic code to tell the ribosomes which protein needs to be made. Transfer RNA, or tRNA in short, is made in the nucleolus. It's made in the nucleolus, not the nucleus. The nucleolus is a part in the nucleus where tRNA and rRNA are made. If you can still remember from lesson 1. From lesson 1, on that video of lesson 1, I said that I will come back to what tRNA is and what rRNA is, and here is me coming back to that and fulfilling my promise. So tRNA is made in the nucleolus, and then it does its functions at the ribosomes. What does it does? Transfer RNA. True to its name, it transfers specific amino acids to the ribosomes to build a polypeptide chain, okay, or to build a protein. Then we have the last type of RNA, which is called ribosomal RNA. I know we haven't spoken about it, but it is quite important for you to know it and know where it is made and where it functions. Okay, it is made in the nucleolus as well. Please mind the spelling, nucleolus and not nucleus. site where it functions in, not at, in the ribosomes. Why do we say in the ribosomes? Because it forms a structural component of the ribosomes. So it is part of the skeleton of the ribosomes. It is part of the scaffolding of the ribosomes. Okay, so it is a component, a part of the ribosomes. Hence, it is found in the ribosomes, not at the ribosomes, like tRNA. Okay guys, you know that assessment is a very important part of the learning process. So here is a third activity on this lesson. Okay, right. So this activity forms part of the amino acid coding, guys. Forms part of the amino acid coding. Okay, remember we said that the lesson, it is going to be a two-part lesson. And the first part was describing the process of protein synthesis, but here we are looking at amino acid coding. Okay, now remember protein synthesis occurs in the following ways. Firstly, we have a DNA code. Okay, and then we have a DNA code, right? And during transcription, we are making mRNA, and then the code within the mRNA is called what? It is called a Why am I writing this? These things are there on the table. Sorry for wasting your time. Okay. There. We have a DNA code. Remember, for which protein is going to be made? The message comes from DNA. So DNA is the instructor that instructs the cell which protein is going to be made, which amino acids must be packed in what order. That instruction comes from the genetic code that is found on the DNA. So during transcription, this DNA code is transcribed into an mRNA codon. Remember what we said the role of mRNA is? mRNA takes the DNA code and takes it to the ribosomes, where tRNA must come and bring a specific amino acid according to its anticodon that is complementary to the codon of the mRNA. So with amino acid coding, we start from the DNA code, then the mRNA codon, and then the tRNA anticodon, which codes for a specific amino acid. All right, so for that we need to do this activity. So here on this table we have a codon, we have this over there, we have this step, we have an mRNA codon on this table. Okay, and then on the right hand side we have the amino acids. So for example, the mRNA codon GGC codes for glycine, the amino acid glycine. AGA codes for the amino acid methionine. The codon UCA codes for the amino acid leucine, and GCC codes for the amino acid glycine. And then we have CAG, which codes for the amino acid tryptophan, and UUU codes for the amino acid lysine. Remember I said you don't need to memorize the names of the amino acids. You will always be given the amino acids in the form of a table. So the first thing that you need to do when you answering the questions of amino acid coding, check the table that you are given and what information does it contain. It will always contain amino acids, guys. It will always contain amino acids. However, on the left-hand side, check what it has. It may have the DNA code, and then on the right side, it shows you which amino acid is coded for by that DNA, or the table may contain the tRNA anticodon there, but in this instance, it is having the codon, and the codon, we know it is the 3 consecutive nitrogenous bases that are found on the mRNA. Okay, use the information from the above table to complete the table below. Okay, now let's check. From our DNA, where is our question 1.1.1? Our question 1.1.1 is there. The DNA code is AGT, which will be complemented by U bonds with A, C bonds with G, and A bonds with T. Now, question 1.1.1, we need the anticodon. We need the anticodon. We need to code for this anticodon. So we already have the mRNA codon, which is UCA. What is the complementary anticodon of UCA? Now, U bonds with what? U is complementary to A, right? And then C is complementary to G, and then A is complementary to U. So the answer there is supposed to be UGA. Let's check the answer side. Is it UGA? Absolutely it is. Now we move to question 1.1.2. The DNA code is AAA. The complementary mRNA codon will be UUU. And then the tRNA anticodon is AAA. This question now is seeking for the correct amino acid. Now, let us check. What do you have there on your table? Your table contains the codon. So it contains that part. So the codon, you just go to your table. The codon is UUU. And then you go to your table to check if you have a UUU. UUU is there. And it codes for the amino acid what? It codes for the amino acid lysine. Okay, guys? So lysine is the correct answer, as you can see over there. Let us now move to question 1.1.3. Question 1.1.3, you are given nothing on the DNA code. You are given nothing on the DNA code. However, you have CAG on your as your mRNA codon. So if you have CAG, it means that you must code up to DNA. But be very careful. Remember, the coding from DNA to RNA differs from the coding. Okay, the coding between DNA and RNA differs because remember, DNA contains thymine and RNA contains which nitrogenous base? Uracil. So here we need to code up to DNA. So what is complementary to C? That would be G. What is complementary to A in DNA? That would be T and not U. And then what is complementary to G in DNA? That would be C. Okay, so my DNA code is supposed to be GTC. Is that the correct answer? Absolutely. Now, question 1.1.4, we need to code down to find the tRNA anticodon. Okay, from this RNA codon. Okay, now let us code. Remember, the complementary base pairing is different because tRNA is an RNA, does not contain T, contains U. So what is complementary to G? to C, it will be G. What is complementary to A in RNA? It is U, not T. And then what is complementary to G? It will be C. So the tRNA anticodon should be GUC. That is correct. Okay, guys? Now, moving from 1.1.4, we go to 1.1.5. We are given the DNA code as CGG. And then the question wants everything else. It needs the mRNA codon, the tRNA anticodon, and the corresponding amino acid. Okay, guys? So, let us code. It means we have to code 1, 2, 3 down. So, what is complementary to CGG? It would be GCC. Okay? For our mRNA codon. GCC is correct. And then, after we have found the codon, we need to check the tRNA anticodon. What is complementary to GCC in terms of tRNA anticodon? It will be C, G, and G. Are we correct? Let's check on the answer portion. That is correct. CGG. And then we need to find the amino acid. We need to find the amino acid. Remember, how did we say we find the amino acid? You go to your table. Your table has which information? Your table has the codon. Now, we need to check the codon. We need to check the codon. The codon is there. There is our codon as GCC. You go back to your table. Do we have a GCC? We do have a GCC. And there is our GCC. And then the relevant codon there. Not codon, sorry. The relevant amino acid there would be glycine. So the answer to question 1.1.7 is glycine. Moving on to question 1.1.8. 1.1.8 is void, 1.1.9 is void, 1.10 is also void. But here we are given the amino acid methionine. We are given the amino acid methionine. So we are given methionine, we go to the table. There is methionine. Okay? So methionine has the codon, it has the codon AGA. We fill A G A there on the codon of our table. Okay, so once we have AGA as a codon, we will be able to code for the tRNA anticodon and the DNA code over there. Which one should we do first? Let's just go with the anticodon. What is complementary to A? It is U. What is complementary to G? It is C. And what is complementary to A? It is U. So our anticodon is UCU. Let us check. Now we are coding for the DNA code. What is complementary to A? It is going to be T on DNA. What is complementary to G is C. And what is complementary to A? It is T. Okay, guys? So from here, we were given the amino acid methionine. We go to the table. We check methionine. The codon is AGA. We write AGA there on the mRNA codon. Once we have AGA, we code it for the DNA code and we code it for the tRNA anticodon. Okay, now let's check if our answers are correct. So 1.1.8, we are supposed to have TCT. We have TCT. 1.1.9 AGA. we have AGA, and 1.1.10, we have UCU. Okay, and there is our UCU. All right, now we are going to move on to the next activity on amino acid coding.